So here's what happens is, so I got a bunch of samples basically from pieces of the lung like this. So here's normal lung. I got, basically this is the process of forming that lung tumor. As I was doing this, so what I did was I looked for genes that actually change as the cancer gets worse. Either they go up or they go down. If they go up, what do you think they are? Remember those two things we talked about in cancer? V, what do you think? If it goes up as the tumor develops or as the tumor gets worse, what kind of thing is it? Is it an oncogene or tumor suppressor? Yes. If it goes down, G money, what do you think it is? If a gene, as this dysplasia gets worse and forms a tumor, if a gene keeps getting decreased as this process happens, what do you think that gene's important about? What does it do? As the gene goes down, right? Tumor's getting worse. The doctor's going, oh my gosh, we got to watch this patient. Things are going from bad to worse. And as this process is happening, this gene is getting less and less. What do you think it's doing? Is it protective or is it causing the cancer?
Would it be causing the cancer?
No, right? It's going down as the cancer is getting worse, right? Which means that's probably one of those tumor suppressors. It's one of the cops trying to help things out. So what happens is it's as it's going down, the criminals are getting worse and worse. So it was a tumor suppressor. So here's one that I found. So this is my target for developmental lung tumors was this gene here.
Do we have to do a presentation?
Yeah. Oh, you absolutely have to do a presentation. I even might record them too.
About what? Anything you want or just the breast cancer?
No, you got to pick something in breast cancer. So here's why I'm presenting this data to you, is that what I want you to do is give you an example of what I want you to present. Okay?
You know what? Can we do it like a group?
We'll see. Okay. So this is polo-like kinase 1. This is my suspect that I found that was important in the development of lung tumors. As you would imagine, if it's important in cancer, it's definitely important in the cell cycle. This is the trigger for G2 to M transition. So that's metaphase, right? So this is a G2. This is when the DNA gets doubled.
Yeah.
And the chromosomes segregate. I think so. It's been a long time.
Metaphase when you line up.
Yeah, they line up, but this is G2 to M. So there's G1, M, and then, so M in this case is mitosis, not metaphase.
Oh.
Okay. So it localizes to the chromosome. So if the cell wants to divide, it uses this gene to make the cell proliferate. So here's what happens if you lose this protein, you induce pro-apoptotic pathways. That means the cell dies and you inhibit the cell growth. So whatever it's needed, whatever it is, it's absolutely required for the cell to keep dividing. Okay? Now in tumor genesis, here's the cool thing, is that what they found is PLK1 can inhibit transactivation and pro-apoptotic functions, pro-death functions of P53. Remember, P53 is like the big cop. This thing is inhibiting the cop. Is this a tumor suppressor or an oncogene, you think?
I'm thinking tumor suppressor.
Is it suppressing the tumor or is it causing it?
It's causing.
That's right. So it's an oncogene. Totally.
Okay.
Just so that you're all aware, none of these genes start out bad, right? We need cells to survive or to divide in our body. We need to keep replenishing all these cells. So this gene is actually important. What happens is something happens where it's no longer under control. Okay? Here is why I was so interested in this, and this just kind of goes back to that whole criminal history we were talking about for those that were at that class on Monday, is that the expression of this, not only do I think it's important in lung cancer, but it also has been elevated or shown to be highly expressed in all these different kinds of cancers. I mean, there's like one, head and neck, esophageal, gastric, melanomas, ovarian, endometrial. This is a criminal. All right, so this is my candidate. This is the criminal that I was going after, polo-like kinase. Not only is it associated with things that I know are important in cancer, but it's also been found in lots of cancers. Not only that, but it seems to go up in all these different cancers, which makes me think it's an oncogene. And here's what you guys are going to do is we are actually, so when you actually look at the genes, and I'm going to give you this software, it's called IPA. It's awesome. It is the most powerful biological software on the planet. And probably maybe 5% of scientists even have access to it. So you guys are having access to something most scientists don't even have access to. So you throw these genes in, what it does is it tells you all these gangs that are in this list of genes that you put in. Sure enough, the ringleader of my gang, polo-like kinase, its gang comes up in genes that change as the cancer gets worse and cancers that remain really, really bad, that don't get better at all. We can actually, I'm going to show you how to do this, is you can pull up that pathway. So this is like PLK's gang. You can see PLK right here, right? All of these biological processes are involved with separation. It's basically mitosis. And these are all the different genes that are associated with making that cell reproduce or basically proliferate. All of the genes in red I found in this study that actually went up as the cancer got worse. Not only do I find the criminal, but I find all his buddies. All of them here that are colored. Not only that, but then I find weak points in his gang. There's this gene here, it's called SLK, and I can't remember what it stands for. But when I look it up, and this is what you're going to do with this data that we're going to mine for breast cancer, when I look it up, SLK induces PLK, this gene here. What if I take this buddy out? Then maybe this gene never gets that signal. Or there's this gene here, it's called pituitary tumor transforming growth factor or something like that. It's got a long name. Anyway, it's bad. It basically prevents this complex from breaking down. And this complex basically pushes the cell into mitosis. If we knock this gene out, maybe we can just get rid of all this stuff. That's kind of what I'm doing is not only are you just finding one gene, but you're basically kind of describing its environment. It's like with our NFL data, right? You know, we talk about the Broncos, or we talk about the Raiders. If you just say they're good or bad, that doesn't mean anything. You say, hey, the Raiders are bad because they have this player and he does this and their quarterback doesn't do this and this and this. Now you're actually doing an analysis, and that's kind of what we're doing with these genes. Think of these genes like PLK as the quarterback and this is its team. I know PLK is basically going to win cancer because all of its buddies, all of its team are really, really important in that process. All right. So we are looking at breast cancer.